So this is Dr. Monisha here. Okay, so to briefly introduce about myself, I have completed my PhD from IIT Hyderabad, mostly focusing on the cancer biology research. And then I completed my master's from the same institute and UG BTech from Anna University, India campus, Chennai. Okay. So having briefly introduced about myself, in today's session, we'll be discussing about very important NEET questions. Okay, so let me share this. Okay, ma. So let us start the session. So today's first question that we will be discussing is NEET 2020 question. Okay, so the first question that is asked in the NEET 2020 is: in moon being resistance to yellow mosaic virus and powdery mildew were brought about by. Okay, so the options given were hybridization and selection. Option 2 was mutation breeding. Option 3 is biofortification, and option 4 is tissue culture. Okay, so we know that developing disease resistant plant is a very important target so that we can improve the yield of biological crops, right? So in order to make the plants okay resistant, okay, moon bean is in Tamil we call it as pachapayir. Okay, ஸோ அந்த பச்சை பயிர் செடியை வந்து வைரஸ்க்கு ரெசிஸ்டண்டா உருவாக்குறதுக்காக we need a technique, and that technique is called as mutation breeding. Okay, so mutation breeding is a very commonly used technology for creating plants or crops which will be resistant to virus, especially the yellow mosaic virus. Okay, so in the picture you see this white dots, right? These dots are nothing but the virus that has infected the leaves. Okay, so the correct option for creating plants resistant to virus is option 2, which is mutation breeding. Okay, right. The second question, okay, that was being asked in the last year NEET is cocoa alkaloid, which is also commonly called as cocaine, okay, is obtained from which of the following four plants? Okay, so the options given were option 1, which is datura, then option 2 is Papaver somniferum, option C is Atropa belladonna, and option 4 is Erythroxylum coca. Okay, so these are the four plants that were given from which you can actually derive alkaloids, or from all these four plants you can derive alkaloids. But the question was very specific for coca alkaloid. Okay, so in the picture, the plant that you see here is nothing but Erythroxylum coca. Okay, this is the plant, and from this particular plant only we will derive coca alkaloid. Okay, so you can also make a smart guess. Okay, how you can make a smart guess? You see the word cocoa alkaloid, right? So coca is the compound from which you are going to derive from the plant. So and the coca வந்து we are going to get it from a plant which is called as Erythroxylum coca. Okay, so coca is derived from a plant called as Erythroxylum coca, and this particular plant is actually native for South America. Okay, mostly this is commonly found in South American countries. Okay, so please remember cocaine. Okay, cocaine is a drug which is commonly used even to treat cancer. Okay, so this particular cocaine is derived from Erythroxylum coca. Please remember the plant name. Now, next is a very tricky and smart technical question which I actually liked. Okay, so this question number 3 is given below are 4 statements pertaining to separation of DNA fragments using gel electrophoresis. Identify the incorrect statement. So you have to find out the incorrect statement regarding DNA electrophoresis. Okay, so we know that agarose gel electrophoresis is a very commonly used technique for separating DNA based on the size. Okay, so உங்களுக்கு ஞாபகம் இருக்கும், நீங்க படிச்சிருப்பீங்க, அது என்னன்னா DNA செப்பரேட் நம்ம மாலிகுலர் வெயிட் பேசிஸ்ல செப்பரேட் பண்றதுக்காக நம்ம ஒரு டெக்னிக் யூஸ் பண்ணுவோம். ஸோ அந்த டெக்னிக் பேரு gel electrophoresis. In the picture நீங்க பாத்தீங்கன்னா the pointer I am showing, this is nothing but it's a technique which is called as agarose gel electrophoresis, and அந்த டெக்னிக் நம்ம எதுக்கு யூஸ் பண்ணுவோம்னா DNA DNA வந்து size basisல separate பண்ணும். Okay, so larger the size of the DNA molecule, okay, slower will be the movement. So in the kuti kuti bands, what you see at the bottom is the small size DNA fragment, whereas the larger size DNA fragment will be actually more towards the top. Okay, so simple thing that you should remember is, if you want to separate DNA molecules, you will use a technique which is called as gel electrophoresis. Okay, so whenever you see gel electrophoresis, it is nothing but a technique to analyze DNA segments. Okay, so having known about that particular point, let us first figure out which is going to be the correct options given in the following four statements. Okay, so the first statement is DNA is negatively charged molecule, and so it is loaded on gel towards the anode terminal. Okay, so if you remember, what is this thing? DNA is of course a negatively charged molecule. Why is DNA negatively charged? Is because DNA molecules have phosphate. Okay, PO4 2 minus. The 2 minus will actually give you a negative charge. Okay, therefore this particular statement, which is the first statement, is actually a correct statement. Okay, so the option 1, which is the DNA is a negatively charged molecule, is of course the right option. Now let us read the next one. What is the next one says? DNA fragments travel along the surface of gel whose concentration does not affect the movement of DNA. Okay, so this particular technology of separating the DNA is absolutely based on the molecular weight. Okay, so it does not have anything to do with the concentration. Okay, therefore concentration does not affect the movement. So this is also a correct statement. Okay, the third statement, the smaller the size of the DNA fragment, larger is the distance it travels through. Okay, so of course, whenever the size of the DNA is going to be small, then the movement of the molecule will also be faster. Okay, next question is, pure DNA can be visualized by exposing to UV radiation. So if you remember, in order to absorb the DNA under the UV radiation or UV light, you need a dye. Remember which is the dye? Anybody remembers what is the dye which we use for staining DNA? Yes, in the recombinant chapter you would have read. Yes, Govardhan, do you know how can you see the DNA when you actually submit? Yeah, what is the dye used? It's called as ethidium bromide, right? So ethidium bromide, yeah, very good. So ethidium bromide will give you an orange color when you actually look under UV radiation. Okay, so that is also the right answer. So the incorrect option, what you can actually see here is the C. Okay, so C is the incorrect answer. Therefore, if you choose 1, 2, 3, 4 option, the option 1, okay, the right option is option 1. Okay, so the summary that you have to take, message that you're going to take from this particular question is, DNA can be separated by a technique which is called as agarose gel electrophoresis, and DNA is a negatively charged molecule because it has a phosphate group. Okay, and usually the DNA can be visualized under the UV light, okay, by using a dye which is called as ethidium bromide. Okay, right. Now, next, let us move on to the fourth question. Okay, the fourth question is, an enzyme catalyzing the removal of nucleotide from the ends of DNA is. Okay, there are 4 options which were given. The first option is protease. The second one is DNA ligase. Third option is endonuclease, and fourth option is exonuclease. Okay, so before we actually see what is the right answer, we know that nucleases are nothing but enzymes which are capable of degrading nucleic acids. Okay, நமக்கு தெரியும் DNA is made up of nucleic acids, correct? DNA is a polymer of nucleotides, right? So it's actually called as deoxyribonucleic acid. It's a polymer of nucleotides. So when you have to degrade, or when you want to remove the nucleotides from the DNA, there are specialized enzymes which are called as nucleases. Okay, so the first option that is given here is something called as proteases, right? What is proteases? Proteases are the enzymes which will degrade protein. Okay, so therefore proteases is an incorrect answer. Similarly, if you remember DNA ligase is an enzyme. What is the function of DNA ligase? It helps in sealing the DNA. right? It's also called as molecular glue. Okay, so DNA ligase is actually a molecular glue, therefore that is also an incorrect answer. Third one is very tricky. Okay, so third one is endonuclease and fourth one is exonuclease. So nucleus வந்து ஒரு enzyme. அந்த enzyme என்ன பண்ணும்? DNA degrade பண்றதுக்காக யூஸ் பண்ற enzyme. அந்த nuclease வந்து two types. ஒன்னு வந்து endonuclease இருக்கு, இன்னொன்னு வந்து exonuclease இருக்கு. Okay, so நம்ம வந்து எந்த enzyme actually யூஸ் பண்ணுவோம் அப்படின்னா exonuclease தான் recombinant DNA technologyல யூஸ் பண்ணுவோம். Okay, so now let us see what is the difference between exonuclease and endonuclease. Okay, so in the picture like you see, exonuclease வந்து it is a specialized enzyme, okay, which can actually remove nucleotides or it can remove the nucleic acid from the terminal site. கார்னர்ல மட்டும்தான் வந்து டிஎன்ஏ ஃபிராக்மெண்ட்ஸ் அதால எடுக்க முடியும். So can you see from the leftmost corner DNA fragment வந்து it is removed by exonuclease. Okay, whereas when you see endonuclease, what is endonuclease? Endo means it means within, right? So endonuclease என்ன பண்ணும்னா DNA fragments in between ல இருந்து நடு போர்ஷன்ல இருந்து கட் பண்ணி எடுக்கும். Okay, so rDNA technology இல்ல cloning நம்ம பண்ணும்போது we don't use endonuclease, we only use exonuclease. Okay, so if you get a question like this, that which of the following is used for removing the DNA from ends? Okay, so அதுதான் நீங்க ரொம்ப கேர்ஃபுல்லா பார்க்கணும். From the ends, from the corners ல இருந்து DNA நம்ம remove பண்ணனும்னா we use a enzyme which is called as exonuclease. Okay, endonuclease வந்து middle of the portion of the DNA ல இருந்து கட் பண்ணி எடுக்கணும். Whereas exonuclease வந்து terminalsல இருந்து இல்ல endsல இருந்து it will remove the DNA. Okay, protease வந்து proteins degrade பண்றதுக்காக யூஸ் ஆகிறது and ligase is for joining the DNA. DNA வை கட் பண்றதுக்கு nuclease யூஸ் ஆகும் and DNA வை join பண்றதுக்கு we use ligase. Okay, I hope it's clear. So Govardhan, is it clear now? Is it clear for you?
Yes, ma'am.
Yeah. What is protease used for? What is what is a protease enzyme? It can degrade proteins. Very good, excellent. And what is endonuclease do?
Cutting the center of the nucleus.
Center, very good. And how about your exonuclease?
Cutting the first side of the nucleus.
Yeah, it is from the ends, either from the right side ல இருந்தோ இல்ல left side ல இருந்தோ. ஆனா வந்து என்ன ஆகும்னா endல இருந்து தான் வந்து அதால DNA வை it can remove. Okay, very good, excellent. Right. Now next one, the fifth question that we are going to discuss today is in RNAi, the genes are silenced using. Okay, this is a NEET 2019 question. Okay, so there were four options given. DS அப்படின்னா double standard. Okay, and SS அப்படின்னு பாத்தீங்கன்னா single standard. Okay, so in the picture, in the last pictureல பாத்தீங்கன்னா there are two strands, right? இது மேல இருக்கிறது ஒரு strand and கீழ இருக்கிறது இன்னொரு strand. Okay, so whenever you see the question DS means double standard. Okay, so the options given were double standard DNA, double standard RNA, single standard DNA, single standard RNA. Okay, so RNAi na enna first? RNAi is nothing but RNA interference. Okay, so usually இப்போ நம்ம பாடியிலே நிறைய genes இருக்கு இல்லையா? Our body is comprised of many genes. For example, we have insulin gene, we have thyroid genes, then we have keratin gene, growth hormone genes, and then உடம்புல நிறைய genes இருக்கு. Okay, so அந்த genes வந்து நம்ம கண்ட்ரோல் பண்ணணும், ரெகுலேட் பண்ணணும் அப்படின்னா, நம்ம அதோட லெவல்ஸ் வந்து கம்மி பண்ணணும் அப்படின்னா, there is one technology called as RNAi. Okay, so RNAi stands for RNA interference. Okay, so RNA interference நம்ம என்ன பண்ணுவோம்னா நம்மள பாடியில இருக்க ஜீன்ஸோட எக்ஸ்பிரஷன்ஸ் வந்து நம்ம கம்மி பண்ணுவோம். Okay, so RNAi technologyல எந்த molecule of nucleic acid is being used is the question. Okay, so அதுல four options இருக்கு. double stranded DNA வா RNA வா இல்ல single stranded DNA வா இல்ல single stranded RNA வா is the question. Okay, so the answer to this particular question is double stranded RNA. Okay, so double stranded RNA வந்து நம்ம RNA interference-க்கு we are using. Okay, so in the picture if you see, actually this is the molecule, the blue color thing that is that you see in the picture is nothing but a double stranded RNA molecule. And RNA molecule will enter into the cell and inside the cell it will block all the RNA molecules. We know that RNA will undergo translation to produce proteins. So நம்ம உடம்புல இருக்க RNA molecules level கம்மி பண்றதுக்காக we use RNA interference, and RNA interference க்கு what molecule we use? Double stranded RNA. Okay, so RNAi question பார்த்தீங்கனாலே you should blindly tick. It's mostly it is double stranded RNA. Okay, so next question is, which is the most common type of embryo sac in angiosperms? Okay, so we know that high angiosperms are going to be the high end variety of plant kingdom, right? So avanga vande we also call them as the flowering plants, you know, because they reproduce by producing flowers. Okay, so there are four options that is given to this particular question, that is bisporic with two sequential divisions, tetrasporic with one mitotic stage of division, third is monosporic with three sequential mitotic divisions, and fourth is monosporic with two sequential mitotic division. Okay, now let us see what is the answer for this. Okay, so the most common way of actually reproducing or to produce embryo sac in angiosperm is by a technique which is called as monosporic. Okay, so once it is going to be monosporic, it will undergo 3 rounds of mitotic division. Okay, so இது நம்ம வந்து சிம்பிளா வந்து explain பண்ணலாம். So in the picture what you see here is MMC. Okay, so MMC is Madras Medical College. So நீங்க வந்து நல்லா focus பண்ணீங்கன்னா MMC லே சீட் வாங்க. Okay, so keyword is MMC. So here MMC stands for megaspore mother cell. Okay, so MMC is nothing but megaspore mother cell. So நமக்கு எப்படி வந்து females வந்து eggs are important for reproduction, similarly in plants also they have this particular MMC which is very very important for producing embryos. Okay, so first round la என்ன ஆகும்னா the MMC which is called as the megaspore mother cell will undergo meiosis. Okay, so if you remember, meiosis is a technology or it is the technique by which a single cell will undergo division, 2 rounds of division to produce 4 daughter cells, right? So each will be haploid in configuration, that is they will have only half the number of chromosomes. So one MMC வந்து it will undergo cell division which is meiosis to produce 4 daughter cells. Okay, so in the picture you see 4 small circle, that means they have produced 4 cells. So one mother cell has undergone meiosis to produce 4 cells. Okay, if you remember the concept, in case of angiosperms, whenever there is this development of embryo sac, out of the 4, 3 of them will be degenerate. What is degenerate? Degenerate means they will not have any ability to produce offsprings. They are just dead cells, or they are neutral cells, I would say. Okay, only one will be live cell which is capable of undergoing reproduction, which is capable of producing offsprings. So in the oral cell, out of the 4 will actually undergo mitosis. Okay, so mitosis is a process of cell division. It is also called as equation division. ஏன்னா mother cell வந்து 2 cell produce பண்ணும்போது each and every cell will have equal number of chromosomes. Okay, so here the single cell will actually undergo mitosis to produce first round of mitosis to produce 2 cells. This particular 2 cell will again undergo second round of mitosis to actually produce 4 cell, and this particular 4 cell will further develop into embryo sac. Okay, so if you see the question carefully, the question says that it's going to be monosporic because you actually develop from single spore or from a single cell. And how many mitotic division is it undergoing? 3 rounds, 1, 2, 3. Okay, so angiosperms la embryo form பண்றதுக்காக you need to have monosporic condition with 3 rounds of mitosis. So finally என்ன ஆகும்னா you will form the embryo. Okay, so remember, in case of angiosperms you have to have a monosporic condition, and under monospores 3 rounds of mitosis undergo பண்ணாதான் embryos can be formed. Okay, so the correct option for development of embryos in angiosperm is monosporic with 3 rounds of mitotic division. So the correct answer is option 3. Okay, so here they have given 2 rounds of sequential division. So 2 rounds is also not enough. அதே மாதிரி இதுல பாத்தீங்கன்னா பைஸ்போரிக் அண்ட் டெட்ராஸ்போரிக் சொல்லியிருக்காங்க. பைஸ்போரிக் கிடையாது, பிகாஸ் இந்த 4 செல்ஸ்ல இருந்து ஒரே ஒரு செல்க்கு மட்டும்தான் லைஃப் கொடுக்குற கேப்பபிலிட்டி இருக்கு. Okay, that's why we call it as a monosporic condition. Okay, bisporic na rendu spore irukkum, rendu spore is not the right one. Similarly, tetrasporic na 4 spores are involved, 4 spores me kadiyadu. Okay, only one spore out of the 4 will actually have the capability to give life, okay, or to develop into embryo. And the embryo form பண்றதுக்கு முன்னாடி 3 rounds of mitosis இருக்கு. So the right option is monosporic with 3 sequential mitotic division. Okay, so let's move on to the next question. That is, from the following, identify the correct combination of salient features of genetic code. Okay, so if you remember what is a genetic code, genetic code is nothing but it's actually code which is present in your mRNA molecule. mRNA is nothing but again RNA is made up of nucleotides. Okay, so mRNA, whenever you have a stretch of 3 nucleotides, we call that as a codon, right? So any 3 nucleotides present is called as a codon. So a genetic code is nothing but it's a box or it's a table listing down various codons that are present in RNA. Okay, so this is actually the box showing different codons which will code for amino acid. So you have to remember, okay, it's a very important point that you should remember whenever you study about genetic code. It's a very, very universal table, whether it's going to be with respect to Indians or it's going to be Americans or it's going to be Chinese. All of us are going to have a universal DNA. Okay, mostly whatever DNA and RNA that we are going to have is going to be universal. Okay, of course there are a lot of variations, that's why we are all unique, right? But whenever it comes to genetic codes are always universal. They are unambiguous. Okay, unambiguous means they are kind of unique ones. Okay, they are not repeating. So if you see you here, you don't see you anywhere in the box. Okay, that it means that it's unambiguous, non-overlapping. Okay, or overlap agadu. Okay, and it is degenerate in nature. It's going to be common for all the organisms that is present in the nature. So in the exam, if you find out what is the feature of genetic code, you have to remember 4 important features for a genetic code. So what are the 4 features? They are going to be universal, they are unambiguous, they are going to be non-overlapping, and they are degenerate. So in the 4 points வந்து நீங்க ஞாபகம் வச்சுக்கணும். Okay, right. Next is a very interesting question asked in NEET 2018. Okay, so this question states that which scientist experimentally proved that DNA is sole genetic material in bacteriophage? So this is a question. So in this question, if you see, there are 4 options. The first option is the Jacob and Monod. The second one is Beetle and Totem. The third one is Messelson and Storm. And fourth option is Hershey and Chase. Okay, so if you remember, there are total 3 important experiments that were performed by scientists to show that DNA is genetic material. Okay, inside cells you have proteins, you have lipids, you have carbohydrates, you have even RNA. Okay, but out of all the biomolecules that you have inside the cell, DNA is the genetic material, or we also call it as a hereditary material, because நம்மளோட next generation க்கு pass பண்ண போற information எதுல இருக்கு? DNA ல தான் இருக்கு. Okay, so DNA வந்து very very important molecule. Otherwise if ஏதாவது DNA ல damage இருந்ததுன்னா what can happen? The person can develop cancer, the person can develop genetic disease, right? So DNA is a very very important material that's present inside the cell, and DNA தான் genetic material அப்படின்னு கண்டுபிடிச்சது யாருன்னா Hershey and Chase experiment தான். So the picture, if you see, this is actually a very nice picture showing how did they actually show or prove that DNA is a genetic material. Okay, so for proving this particular concept of DNA as genetic material, Michelson and Stoll also did certain experiments, but Hershey Chase the final they proved it. Okay, so in the particular experiment, what they did is Hershey and Chase, they took 2 important organisms. One is a bacteriophage. Okay, so anybody knows what is a bacteriophage? Anybody? What is bacteriophage? Ma? You remember this picture that you see here is a bacteriophage, right? So what is bacteriophage? Anybody? Okay, so bacteriophage ஒரு living organism தான். ஆனா bacteriophage என்ன பண்ணும்னா, it's a virus. Okay, and the virus it can infect or it can kill only bacteria. Okay, so bacteriophage means it's a virus that is capable of infecting bacteria. Okay, so in Hershey and Chase experiment, two organisms were used. The first organism is a bacteriophage and the second one is actually a bacteria. So these are the two organisms they used. Okay, so to prove that DNA is a genetic material, they do two sets of experiment. Okay, one set of experiment what they did is they actually labeled the protein. Okay, so protein label பண்றதுக்கு you need capsids. Okay, so they took a radio labeled protein or a radioactive bacteriophage which was labeled for protein. Okay, in another test experiment what they did is they took a bacteriophage that was radioactive for DNA. Okay, so to make a DNA radioactive, you should use a radioactive phosphorus. Okay, so Repside panel, they took a phage or a virus which is actually radio labeled for proteins, and in the second compartment, they took a virus that was actually radio labeled with the DNA. Okay, so when they infected the E. coli cells, E. coli is Escherichia coli. So E. coli cells, when they actually infected with this particular virus, they found that actually the E. coli cells which were infected only with radio labeled DNA become radioactive. Okay, so this is how they showed that DNA is a very very important genetic material, and this particular experiment was performed by Hershey and Chase. Is it clear? So very very important. Please remember, DNA வந்துதான் நம்மளோட செல்ஸ்ல இருக்க genetic material or hereditary carrying information molecule. Okay, so DNA is a genetic material னு prove பண்ண experiment என்னதுன்னா Hershey and Chase experiment using radio labeled bacteriophage and E. coli cells. Okay, so now next let's go on to the ninth question, that is what initiation and termination factors are involved in transcription. Okay, so it's a very very important concept. Okay, so anybody knows what is transcription here? Very very fundamental one. Transcription na enna? Anybody? Govardhan, you know what is transcription, ma? Don't feel scared. If you have any doubts, you are free to ask me. Okay, I'm here to solve your doubts. Okay, right. Do you know what is transcription, Govardhan? Transcription enna theriyuma? Okay, so transcription அப்படின்னா என்னன்னா, நம்ம உடம்புல என்ன இருக்கு? டிஎன்ஏ இருக்கு. டிஎன்ஏ தான் ஹெரிடிட்டரி மெட்டீரியல். கரெக்ட்? இந்த பிக்சர்ல நீங்க பிக்சர் பாத்தீங்கன்னா தெரியுது டிஎன்ஏ. ஓகே, சோ டிஎன்ஏ நம்ம ஆர்என்ஏவா மாத்தணும். Okay, so DNA RNA மாத்துற process பேரு தான் transcription. So it's the process of converting DNA into RNA. Okay, so in the picture the light sky blue that you see here is RNA. So this process of converting your DNA into RNA is called as transcription. Right? Now once you have converted the RNA molecule, RNA can be further converted into proteins. Okay, so when you convert RNA into proteins, there is one more technique which is called as translation. Okay, so translation is nothing but converting RNA into protein. So let me make it very simple. So DNA va RNA va மாத்துற அந்த ஸ்டெப் பேரு transcription. RNA va protein மாத்துறது அந்த ஸ்டெப் பேரு translation. Is it clear, ma? So now tell me what is transcription?
DNA to RNA.
DNA to RNA. Very good. Excellent. Okay. So in the step, if நம்ம ஒரு DNA molecule RNA வா மாத்த போறோம். So அது மாத்துறதுக்காக we actually have three important steps. Okay. And the three என்ன stepsனா initiation, elongation, termination. Okay. So transcription, which is nothing but converting DNA into RNA. So transcription பண்றதுக்காக there are three steps, that is initiation, elongation and termination. Okay, so initiation நமக்கு புரியும். Initiate பண்றது அப்படின்னா என்னது? To start. கரெக்டா? So transcription நம்ம start பண்ற event பேர் தான் initiation. So அந்த initiation, அதாவது நம்ம DNA RNA வா மாத்துறதுக்கு initiate பண்றதுக்கு we required specialized enzymes which are called as RNA polymerase and sigma factor. Okay, so sigma factor is a very very important factor that is involved in initiation. So in the question நீங்க கேர்ஃபுல்லா பாத்தீங்கன்னா, what is the initiation factor? So first question என்ன? Initiation. Okay, so transcription is nothing but converting DNA into RNA. அதுல வந்து மூணு sub steps இருக்கு. என்ன இருக்கு? Initiation, elongation, termination. Initiation தான் first step. அந்த first step transcription பண்றதுக்கு we require a protein or an enzyme which is called as a sigma factor. So இந்த pictureல இந்த green color நமக்கு தெரியுதுல, that's called as a sigma factor. Okay, so sigma factor DNA ஓட bind ஆனாதான் transcription initiate ஆகும். Okay, so that's called as an initiation factor. So initiation ஆனதுக்கு அப்புறம் என்ன ஆகும்னா elongation ஆகும். Elongation, can you see the blue color RNA molecule is growing in length. அதோட size வந்து increase ஆகுது, mRNA molecule வந்து production ஆகுது. Okay, so கடைசியில last step என்னன்னா termination. Termination என்ன? அந்த DNA ல இருந்து RNA முடியிற ஸ்டெப். டெர்மினேட்னா என்ன பண்றது? ஸ்டாப் பண்றது. Okay, so அந்த transcription process வந்து முடிக்கிற ஸ்டெப், the climax step is called as transcription. Sorry, it's called as the termination step. Okay, so transcription is nothing but converting your DNA into RNA, and for this conversion to happen you have three steps, that is initiation, elongation and termination. So termination என்ன ஆகுதுன்னா complete RNA molecule is getting synthesized, and for the termination to happen you have a special factor which is called as the rho factor. So the P மாதிரி தெரியுதுல, the P symbol is actually called as a rho. Okay, and it's the Greek symbol. So rho வந்து it's important for termination. Okay, so transcription is converting DNA into RNA, and this particular conversion requires three steps: initiation, elongation and termination. Initiate பண்றதுக்காக யூஸ் பண்ற factor is called as the sigma factor, and terminate பண்றதுக்காக யூஸ் பண்ற factor is called as your rho factor. It's like a P symbol. Okay, so the right option for this is, now tell me what is the right option? Govardhan?
Option 2.
Option 2. Right? What is the function of sigma? Sigma factor. What is the use of sigma factor?
Initiation process.
Very good, for initiation. And what is the function of rho factor?
For termination.
Termination. Okay, so transcription is a process of converting your DNA molecule into RNA, and for this entire process you require two important components, that is the sigma factor, which is initiation, and rho factor, which is the terminator, or it's a kind of a climax for transcription. Okay, very good. Right. Now next one is another interesting question. Okay, so in which genetic condition each cell is in the infected person has 3 sex chromosomes? So usually how many sex chromosomes a human cell will have? Anybody?
23
pairs.
23 pairs, right? The 23rd pair, if it is going to be XX, then what is the sex of the person?
Female.
Female. If you have XY, what is the sex?
Male.
Male. Very good. So that is a normal process, that if it is XX, it's female. If you have XY, it is going to be male. And there are few genetic disorders, okay, where you have extra X. Okay, that is the person will become, it's supposed to be a male. Okay, so XY they are supposed to be a male, and they have an extra X copy, because of which they will develop female characteristics, like they'll have enlargement of breast. Okay, usually males will not have these secondary sexual characteristics, but in XXY condition what will happen is the male will develop feminine characteristics. So this particular condition where you have XXY is what disease is the question. So when you're going to be doctors in future, you have to remember all these disease names. Okay, that is like what is Turner syndrome, what is thalassemia, what is Klinefelter syndrome, and what is phenylketonuria. Okay, all these four are actually genetic disorders. Phenylketonuria is a condition where you have deficiency or metabolism error in phenylalanine. Okay, so phenylalanine is an important amino acid. So and the amino acid deficiency they will develop phenylketonuria. Okay, அதே மாதிரி thalassemia. Thalassemia is a condition where you have a mutation in RBC. Okay, what is RBC? Anybody?
Red blood corpuscles.
Red cells. Okay, normal red blood cells it will be red in color. It is a biconcave disc shaped structure. Correct? Proper spherical. And thalassemia என்ன ஆகும்னா the person will have mutation in RBC. அதனால what will happen is the person will develop anemia. Okay, நம்ம உடம்புல even in females, when we lose lot of blood, when our HB count is very less, then what we call, we have anemia, right? So thalassemia is an disease associated with reduction in RBC leading to anemia. Okay, so the right option for this particular thing is Klinefelter syndrome. Okay, so in case of Klinefelter syndrome, what will happen in this picture, you see he or she, right? He actually, he will be having a additional copy of X. Okay, so XXY. Okay, in exam, if you see XXY, it is nothing but it is Klinefelter's syndrome. Is it clear? Right, very good. Now, the 11th question that we'll be discussing in today's session is, in the process of transcription in eukaryotes, the RNA polymerase transcribes option 1 precursors of mRNA, hnRNA, option 2 mRNA with additional processing, capping and tailing, option C tRNA, 5 sRNA and snRNAs. Option 4 is rRNAs, 28S, 18S and 5.8S. Okay, so let us discuss about this question in detail. So first one, let us understand what this question means. Okay, so in the process of transcription in eukaryotes, now we have discussed in the last slide that what is transcription. So tell me, what is transcription?
DNA to RNA.
DNA to RNA. Very good. So in this picture, you see the DNA is getting converted into RNA. So that is called as transcription. Okay. So transcription pandrathukaga, what are the 3 steps involved in transcription?
Initiation,
Very good. Initiation,
extension,
elongation, yes,
and
and
termination.
termination. So in the 3 steps, initiation, elongation, and termination are the 3 important steps to perform your transcription. And transcription is nothing but DNA to RNA. So in the step, DNA வந்து RNA மாற்றத்துக்காக யூஸ் பண்ற enzyme is called as RNA polymerase. So let me go back to this picture. So here, this green color that you see here is nothing but RNA polymerase. So RNA polymerase is an enzyme which creates RNA molecules. Okay. So in this question, RNA polymerase transcribes which of the following components? Okay. So here the right option is rRNA with 3 subunits, that is 28S, 18S and 5.8S. So very important. Now, eukaryotes வந்து, what are the different types of RNA? Anybody knows?
mRNA, tRNA, and rRNA.
Very good, excellent. Okay, so there are 3 types: mRNA, tRNA, and rRNA. So in the question, நீங்க carefully பாத்தீங்கன்னா, they are asking RNA polymerase 1. Okay, so RNA polymerase 1 ஓட function என்னன்னா, to produce RNA molecules, that is rRNA molecule. rRNA na ribosomal RNA. So polymerase 1 அப்படின்னு நீங்க questionல பாத்தீங்கன்னா, the answer is rRNA. அதே நீங்க question வந்து polymerase 2 அப்படின்னு பாத்தீங்கன்னா, the answer is going to be tRNA. tRNA means transfer RNA. Okay, அதே நீங்க questionல polymerase 3 அப்படின்னு பாத்தீங்கன்னா, the answer is going to be mRNA. Okay, so there are 3 different types of RNA molecules that are present inside our cell, that is mRNA, tRNA and rRNA. And the 3 different types of RNA molecules produce பண்றதுக்காக we have 3 different types of enzymes, that is RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. RNA polymerase 1 அப்படின்னு examல பாத்தீங்கன்னா, the answer is going to be rRNA. Polymerase 2 அப்படின்னு பாத்தீங்கன்னா, it is tRNA. Okay, polymerase 3 அப்படின்னு questionல பாத்தீங்கன்னா, it is going to be mRNA. Okay, so இது வந்து additional statement வந்து உங்களுக்கு just confuse பண்றதுக்காக கொடுக்குற statement. Okay, nothing to do with 28S. Of course, these are the molecular weights of the RNA. That is 28S, 18S and 5.8S. Anybody knows what is the S unit that is mentioned in the question? What is S stands for? S is actually Swedberg unit. Okay, so Swedberg unit வந்து எதுக்கு யூஸ் பண்றாங்கன்னா, RNA molecules oda molecular weight represent பண்றதுக்காக. For example, if you have say for example you have NaCl. Okay, so NaCl oda molecular weight வந்து நம்ம என்ன சொல்றோம்? Say 43 அப்படின்னு சொல்றோம். எப்படி நம்ம சொல்றோம்? What unit we represent? We represent it in Daltons, right? So molecular weights of usually chemical compounds are represented by Daltons. Whereas in case of RNA molecules, we actually represent the molecular weight in terms of a unit which is called as a Swedberg unit. And இது வந்து just additional statement உங்களை confuse பண்றதுக்காக they have given. Okay, so you have to make sure that what is the question they're asking about transcription. So transcription is nothing but DNA to RNA. Ana human cell there are 3 different types of RNA, that is mRNA, tRNA and rRNA. mRNA produce பண்ணனா polymerase 3 வேணும், tRNA produce பண்ணனா RNA polymerase 2 வேணும், and rRNA produce பண்றதுக்கு தான் polymerase 1 வேணும். So the right option is rRNA. Okay, so can you tell me what is the enzyme that is used for producing mRNA?
Polymers.
RNA polymerase 1.
RNA polymerase?
One.
mRNA. mRNA for mRNA.
3, ma'am.
Not 3. Very good. tRNA ku
2.
2 and rRNA ku
1.
1.
1. Very good. Okay, so இந்த numbers நீங்க ஞாபகம் வச்சுக்கணும். Polymerase 1 எதுக்கு, polymerase 2 எதுக்கு, polymerase 3 எதுக்கு, and what type of RNA? Is it mRNA, tRNA வா இல்ல rRNA? R na ribosomal RNA, tRNA transfer RNA, mRNA na messenger RNA. ஓகே, சோ இதுல அடிஷனல் ஆர்என்ஏவும் இருக்கு. ஃபார் எக்ஸாம்பிள் இந்த கொஸ்டின்ல பாத்தீங்கன்னா HNRNA அப்படின்னு இருக்கு. இத வந்து ரொம்ப மைனர் ஆர்என்ஏஸ், அதோட ஃபங்க்ஷன்ஸ் வந்து ஸ்டில் பீப்புள் ஆர் டூயிங் லாட் ஆஃப் ரிசர்ச். அதோட ஃபங்க்ஷன் வந்து இன்னும் தெரியல ப்ராப்பரா. HNRNA ஃபார் எக்ஸாம்பிள் இப்போ mRNA, tRNA, rRNA வந்து 99% நம்ம செல்ல இருக்கும். ரிமைனிங் அந்த 1% வந்து HNRNA இருக்கும், இல்ல SNRNA இருக்கு. SNRNA ஸ்மால் நியூக்ளியோலர் RNA. ஓகே, இது வந்து ரொம்ப இன்சிக்னிபிகன்ட் நம்மளோட பாடியில ஃபங்க்ஷன் வந்து. பட் தே ஆர் ஆல்சோ பிரசன்ட் அட் மைனர் லெவல். அதோட ஃபங்க்ஷன் வந்து பீப்புள் ஆர் ஸ்டில் டூயிங் லாட் ஆஃப் ரிசர்ச் டு ஐடென்டிஃபை வாட் இஸ் த ரோல். Okay, right. So this 12th question is actually a very theoretical question. There is not much of logic involved. This is from the evolution chapter. So mostly evolution chapter nale you have to remember theories and facts. Okay, so adla not much logical questions will be asked. So this is a theoretical question. So which of the following statement is correct about origin and evolution of men? So this is from evolution chapter. The first option is Neanderthal men lived in Asia between 1 lakh and 40,000 years back. So when you convert this into millions, anybody, how can you convert this into million? How do you convert 1 lakh into million? How many millions is 1 lakh? What is 1 million? 1 million is 10 power 6, right? That is 10 lakhs is nothing but 1 million. So 1 lakh is equal to 0.1 million. Sometimes you might get the questions in MYA. Okay, what is MYA? Anybody? எவல்யூஷன்ல நிறைய நீங்க MYA பார்த்திருப்பீங்கல்ல புக்ல. MYA னா மில்லியன்ஸ் ஆஃப் இயர்ஸ் அகோ. ஓகேங்களா? சோ இது வந்து 0.1 MYA னு எழுதலாம், இல்லனா 1 lakh னு எழுதலாம். 1 lakh years ago வந்து is nothing but 0.1 MYA. MYA na millions of years ago. Okay, so this particular men, that is Neanderthal men, they actually lived in Asia. Asia means it includes India, it includes your China, Japan, all the Asian countries, Singapore, Malaysia, all these countries and all. Neanderthal men lived, and they actually lived around 1 lakh to 40,000 years back. Okay, so that is actually the right statement about evolution of man. Agriculture came around 50,000 years back. That is again an incorrect question. Then the Dryopithecus and Rampithecus primates existed 15 million years ago. That's again a very false statement, because if you remember, the human population started only 4 million years ago. Okay, so 15 million is a wrong statement. So that is also incorrect. Next is Homo habilis probably ate meat. Okay, so these people mostly they eat only vegetables, fruits, whatever was available. So they ate meat is again incorrect statement. Okay, and here, yeah, I was referring to this MYA, right? So MYA means it is millions of years ago. Okay, so you have to identify the answer one is the correct one, which is Neanderthal men lived in Asia between 1 lakh and 40,000 years ago. Okay, right. Now, next is 13th question. The production of gametes by parents, the formation of zygotes, the F1 and F2 plants can be understood using which of the following representations? Okay, first one is Venn diagram, second one is pie diagram, third one is pyramid diagram, and fourth one is called as a Punnett diagram. It's also called as a checker box. Okay, right. So now let us see what is this Punnett square. Okay, the answer is Punnett square. So if you remember genetics, so you guys have studied genetics in your yeah. So who is father of genetics?
John Gregor Mendel.
John Gregor Mendel. Very good, excellent. Okay, so what are the 3 postulates that Mendel gave? Do you remember?
Law of dominance, segregation
and law
of independent assortment.
Right. So in order to understand the genetics, very good, excellent, Devishri, excellent, very good. So in order to understand the genetics, people or geneticists actually use a representation which is actually called as a Punnett Square. This particular Punnett Square is also called as the checker box. Okay, so usually it's a 4 square box. Okay, where on the left side you actually put, or I can say it's left side. So left side you will have parent 1. On top of that, you'll have parent 2. Okay, because we are all undergoing sexual reproduction, we require 2 parents, right? So when these parents will actually undergo reproduction, they produce offsprings. Genetics is more or less, it's a probability based subject. Okay, what is the probability that a lady will give birth to a female, or a lady will give birth to a male boy? Okay, it's 50% chance, right? That's a probability based 1:1 ratio. Similarly, in genetics also, what is the chance that the offspring produced is going to be homozygous dominant, or it's going to be heterozygous dominant, or it's going to be homozygous recessive? Okay, so before we go get into that, what is dominant? Anybody? What is dominant? Dominant means in genetics, dominant means it's the characteristics, or it's going to be the trait that is going to get expressed, right? So black hair versus gray hair. So black hair is a dominant variety which usually gets expressed. Okay, so it's the one which will actually get expressed in an individual. Okay, so dominant is a superior trait, and recessive is something which is going to be suppressed, and it's going to be an inferior trait. Okay, so usually dominant characters are represented by capitals, and recessives are going to be represented by small letter. Okay, so parent 1 on the left side and parent 2 is represented on the top panel. Okay, so for example, இப்போ parent A, okay, ஒரு மதருக்கு வந்து capital B gene இருக்கு, fatherக்கு capital B gene இருக்குன்னா, ரெண்டு capital B and capital B will combine together to produce an offspring. That offspring will have capital B, capital B. That is, the baby is going to have both the characteristics dominant. Okay, this is called as a homozygous dominant condition. Okay, suppose the suppose the father is capital B, mother is small b. Okay, so in the ரெண்டு gamete fuse ஆச்சுன்னா, the offspring or the baby that is produced can be capital B, small b. That is nothing but heterozygous. Heterozygous na ரெண்டு copy of gene la ஒரு copy is not matching with other copy. Okay, so this is called as a heterozygous condition, and it is going to be dominant. Why it is dominant? Because ஒரு capital B இருக்கு, at least ஒன்னு capitals ல இருந்ததுன்னா the offspring will have the dominant character. So this is a heterozygous dominant condition. This box, if you see, he has one capital B and one small b. Again, this is dominant, but it is going to be a heterozygous dominant condition. And the fourth one is the small, both of them are, recessive characteristics. That is, the small b and the small b can undergo fusion to produce an offspring which will have both of them small b's. So this is homozygous, and it is homozygous recessive. ஓகே, ரெசசிவ்னா அவங்களுக்கு கம்மியான கேரக்டரிஸ்டிக் இருக்கும். ஃபார் எக்ஸாம்பிள், ஒரு பர்சன் டாலா இருக்காங்கன்னா அது டாமினன்ட், ஒரு பர்சன் பார்ப்பா இருக்காங்கன்னா இட் இஸ் ரெசசிவ் கேரக்டர். Okay, so this particular last box is homozygous recessive. So when you are going to represent the probability of distribution of offsprings in genetics, we make a representation, and that representation is called as the Punnett Square. Okay, Punnett Square is also called as a checker box. Okay, this is a commonly used representation by genetists in order to understand the way how the characters are getting distributed. Right. So how many set of contrasting characters did Gregor Mendel study in his experiment?
7 pairs.
7 pairs. Very good. Excellent. Okay. Fine. So now next, let us move on to the 15th question, which is removal of shoot tips is very useful techniques to boost the production of tea leaves. This is because, so whenever you go to tea estate like Ooty and all, okay, what do? They always keep trimming the leaves, right? Okay, why do they actually trim? Even when you go to restaurants or whenever you go to parks, you see there will be a person who will always keep trimming the leaf. Okay, it is actually beneficial. Even at homes, when we have gardens, we trim the leaves. Okay, so what is the benefit or what we should actually trim the leaves is the question. Okay, so the options given are gibberellins delay senescence of leaves. Option 2 is gibberellins prevent blotting and are inactivated. Option 3 is auxins prevent leaf drop at early stages. Option 4 is effect of auxins is removed and growth of lateral buds is enhanced. Okay, so the right option, of course, you can see from the answer itself that the right one is option 4. Okay, the option 4 is effect of auxin is removed and growth of lateral buds is enhanced. Okay, so anybody knows what are the 4 important plant growth hormones? Anybody? There are 4 important
Auxin, gibberellins, cytokines, and acetic acid.
excellent, very good. Okay, so here I have actually made a simplified diagram which will actually summarize what are the important properties of 4 growth hormones that are present. Okay, like how we have different hormones that control our body activities. Similarly, plants also have hormones in order to control. The 4 hormones present in plants are gibberellins, auxins, cytokines, and auxins. Okay, these are 4 important hormones. So let us discuss little bit before answering this question. Let us understand what are the properties and functions of each of these hormones present in the plants. Okay, so first one is control of cell division. Okay, so plant cells வந்து நல்லா divide ஆகிறதுக்கு you require 2 important types of hormones. They are cytokines and gibberellic acid. Okay, so please remember cytokines and gibberellic acid are very, very important hormones, important for cell division. Okay, so repeatedly, even I didn't அந்த matching section இருக்கு இல்லையா, அதுலயும் they frequently asked what is the function of gibberellic acid, what is the function of cytokines, what is the function of abscisic acid. இதெல்லாம் வந்து very, very important. Okay, so whenever you see the question cell division na ரெண்டு hormone யாரு cytokines and gibberellic acid. Okay, next வந்து stomatal closure. Okay, so stomatoclosure எதுக்கு நமக்கு யூஸ்ஃபுல் ஆகுது? during night times photosynthesis have to stop. கரெக்டா? இல்லனா என்ன ஆகும்னா excessive transpirationனால water molecules will be lost and the plant will get dehydrated. Okay, so stomatal closure is very important. So pores, so stomata open and close பண்றதுக்காக you need a regulating hormone which is called as your abscisic acid. So abscisic acid is very important for opening and closing the stomatal pores. Next is development of abscission zone. Okay, so and the leaf and the abscission zones create பண்றதுக்காக you require ethylene and oxalate. Okay, so ethylene க்கு இன்னொரு function இருக்கு. ethylene வந்து it's a plant hormone. அது எதுக்கு யூஸ் ஆகுதுன்னா it's important for ripening of fruits. Okay, for example, you have banana at home, you keep it a little bit greenish in the night. Tomorrow morning you see what happens, it becomes completely yellow in color, right? That is, the fruit has become ripened. So fruits oda ripening kaga very, very important molecule is ethylene. Okay, so usually even in retail shops and all, when they store this particular mangoes and all, if they wanted to make it ripen faster, what they do is they add a chemical which is rich in ethylene, so that artificially you can promote ripening of plants, ripening of fruits. Okay, so mangoes கூட artificially பழுக்க வைக்கிறதுக்காக chemicals add பண்றாங்க அப்படின்னு சொல்றாங்க. அந்த chemicals are nothing but it is actually ethylene rich, so that it promotes சீக்கிரமா பழுத்துரும். That is not good for health, because naturally the fruit should ripen. Okay, so in order to promote the ripening of fruits, you actually need ethylene. So that's also another important point you should remember. Gravitrophism of roots. So gravitrophism is nothing but movement of the plant in response to a stimulus, right? So in response to gravity, roots can also migrate. Okay, so another property when facilitate பண்றது யாரு அப்படின்னா it is auxins. Okay, so here it is mentioned, right? The induction of fruit ripening is very important. So fruit produce ஆனதுக்கு அப்புறம் அது பழுக்க வைக்கிறதுக்காக you need an hormone in plants which is called as ethylene. Okay, and tiny buds ல இருந்து கொஞ்சம் fruits பெருசா ஆகிறதுக்காக you need an enzyme needs an hormone which is called as cytokines. So cytokines are very, very important for maturation of fruits. Okay, அதே நீங்க வந்து fruit bud இல்லைன்னா ஃப்ரூட்ஸ் வந்து ப்ரமோட் பண்ணணும், ஃப்ளவரிங் ப்ரமோட் பண்ணணும் அப்படின்னா you need gibberellic acid. Okay, so gibberellic acid is important for flowering. And next is control of the stem elongation. So stem or elongation height நம்ம கண்ட்ரோல் பண்ணணும் அப்படின்னா auxins and gibberellic acid is important. Okay, so if you see today lot of genetic engineering and tissue engineering is playing a vital role. People are doing a lot of research. Miniature versions of banana and all you have, right? In if you go to this gift shops, people give this small banana plantlets, right? They are nothing but it's genetically engineered banana plants, miniature version. Okay, right. So this particular thing is actually facilitated by controlling or regulating different hormones that are present in plants. So இப்போ நம்ம வந்து டீ லீஃபோட ஷூட்ஸ் எது ட்ரிம் பண்ணோம்னா auxins is removed so that more of buds can be promoted. So that is the reason why you have actually trimming of shoots. Usually, auxins are important for cell elongation and they are present at the shoot tips. Okay, so நம்ம auxins delete பண்ணிட்டோம் அப்படின்னா buds formation வந்து it will be helpful. So that is the reason. Now, next is a very interesting question. One scientist cultured Cladophora in suspension of Acetobacter. Okay, and what he did is he eliminated the culture. That is, he took a cell culture. He took a flask. In the flask, he had two species. Okay, first species is Cladophora and second species is Acetobacter. And then the species he mixed and then he eliminated, means he is throwing light on the culture. Okay, by splitting the light through prism. Okay, he observed the bacteria accumulated mainly in the region of option A, blue and red, option B, violet and green, option C is indigo and green, option 4 is orange and yellow. Okay, so in the picture what you see, the bacteria that you see is actually called as Acetobacter. Okay, so Acetobacter is an aerobic bacteria. So what does aerobic bacteria means?
Requires oxygen for survival.
Oxygen. Okay, very good. So Acetobacters are those bacteria which actually requires oxygen for their survival. And Cladophora. Cladophora, mostly when you see the word flora, it is an algae. Okay, so bacteriaவும் algaeவும் ஒன்னா சஸ்பென்ஷன்ல க்ரோ பண்ணிருக்காரு. So bacteria வந்து Acetobacter and algae is a Cladophora. So when he actually passed the light, he found that he could actually see more of blue and red light. Okay, so the option is blue and red light. Why? Because you have to remember the point that Acetobacter is actually capable of accumulating this particular blue and red light. Okay, so in future in exams, if you see Acetobacter, then remember Acetobacter has a special property of emitting blue and red light. So அதனால he could see a lot of blue and red lights wherever this particular Acetobacter is present. Okay, this question has nothing to do with Cladophora. அது வந்து just உங்களை trick பண்றதுக்காக they have given. Okay, so Acetobacter அப்படின்னு பார்த்தீங்கன்னா this is a specialized bacteria which are capable of emitting blue and red light. So blue and red light தெரியுது Acetobacter is because this particular bacteria has a special property of emitting this particular light. Okay. Now, next, let us move on to the 17th question. That in order to increase the yield of sugarcane crop, which of the following growth regulators should be sprayed? Okay, so they want to increase the yield. Okay, so the options given were cytokines, ethylene, auxins, and gibberellins. Okay, so the right option is gibberellins. Why? Because in what is the edible part in sugarcane? Which part are we interested in sugarcane?
Shoot.
Yeah, the shoot part, the stem part, right? We don't need sugarcane leaf. We don't need sugarcane fruits or buds. There is no as such. The commercial use or the commercial purpose of growing a sugarcane is to get more of its stem portion, right? So, ஒரு farmer வந்து more stem portion இல்ல more sugarcane ஓட productivity increase பண்ணணும் அப்படின்னு நினைச்சாருன்னா he will definitely apply gibberellins, because if you remember this particular point, the stem length, okay, or the shoot length is actually elongated by gibberellins. அதனால if you actually add gibberellins, what will happen is your shoot length or the stem length will actually increase. It will become more thick. It will become more fleshy, so that he can have a better yield. So sugarcane வந்து crop improve பண்ணணும் அப்படின்னா the farmer will actually apply more of gibberellins, artificial or synthetic versions of gibberellins to soil, so that he can improve the productivity of sugarcane. Next, for 18th question, what type of pollination takes place in Valsinaria? Okay, so Valsinaria, anybody knows what is a Valsinaria? Is it a terrestrial plant or is it an aquatic plant?
Aquatic plant.
It's an an aquatic plant. Very good. So whenever you have pollination by water, what do you call that as? Anybody? Pollination in water. There are different types of phyllis, right? If you remember. Yeah.
Hydrophily.
Excellent, very good. It's called as an hydrophily. Okay, so hydrophily is nothing but pollination. If it takes place in water, we call it as hydrophily. And how about pollination by insects like your honeybees?
Ornithophily.
Okay, it's also called as entomophily, right?
Yes, ma'am.
Yeah. Okay, so entomophily is a condition where you actually have pollination facilitated by insects, right? And yeah, so now let us see what type of pollination actually takes place in aquatic plants, especially in Valsineria. The options given in NEET 2019 were the male flowers are carried by water currents to female flowers at the surface. Okay, of course we know that you have to make a smart guess. Valsineria is an aquatic plant, therefore you should make sure that it is somehow going to happen by water. So option 1 is correct. Okay. Option 2 is also somewhere it is talking about water only. Pollination occurs in submerged condition by water. So this is also somewhere tricky. So both of them are with respect to water. You can easily eliminate option 3 and 4 because option 3 is by insect and option 4 is by wind. We know that it's a water living plant. Therefore, insect cannot merge inside water to create pollination. Therefore, that's also not possible. And even in case of wind, it is not possible because it's an aquatic. So you can easily eliminate option 3 and 4. Now you have to make a smart guess between option 1 or option 2. Okay, so here is the one where you have to actually remember the point that Valsinia exhibits a condition which is called as epihydrophily. Okay, so what is this particular epihydrophily? Is usually in case of Valsinia, this female flower will be present on the top of the water. Okay, so on top of the water, you will actually see the female flowers. And male flowers, so can you see the brown ones that you see in the figure? The brown ones are nothing but the male ones. Usually, the male ones will be present submerged in water. So, remember, in Valsineria, females will be present on the top and males will be present in the bottom. And who will produce the pollen grains? Is it male or female?
Male, ma'am.
Male. So, male is present at the bottom. So, therefore, pollen grains will be present at the bottom only, right? These pollen grains which are present in the bottom has to somehow migrate and reach towards the top so that it's available for the female egg to undergo fertilization. Okay, so in case of Valsineria, please remember that the male flowers which are submerged inside the water, they will produce pollen grains, and these pollen grains will actually migrate from the bottom and they will reach towards the top of the female, which is the top part of the water, which is the surface of the water, where the female is present, and there it will undergo fertilization. And what is the female reproductive part called in flower? No, the female reproductive part. It's called as gynoecium, right? And how about the male reproductive structure? What is the name for male reproductive structure? Yes,
Androecium, ma'am.
Androecium. Excellent. Okay, so this is sometimes they may have a trick in the words in between in that way also. Okay, so androecium is present inside, is submerged in case of Valsinia at the bottom, and gynoecium is actually present on the superficial surface of the water in case of Valsinia.